Hello students and welcome. Today we will be talking about the definition and scope of medicinal chemistry. The main objectives of this topic are to understand what is medicinal chemistry, to know its aim and scope in current and future scenario, role of medicinal chemistry in discovery of new drugs. Now, one might ask what medicinal chemistry is and what is its aim. Let us first describe what the field of medicinal chemistry is concerned with. Medicinal chemistry is the chemistry discipline concerned with the design, development, and synthesis of pharmaceutical drugs. The discipline combines expertise from chemistry and pharmacology to identify and develop and synthesize chemical agents that have a therapeutic use and to evaluate the properties of existing drugs. It has a lot of scope in future and current period as medicinal chemistry has evolved rapidly into a highly interdisciplinary field, enriched by the collaborative efforts of experts from a wide spectrum of specialist areas, from chemoinformaticians and physical chemists to molecular biologists and pharmacologists. Medicinal chemistry uses physical organic principles to understand the interaction of small molecular displays with the biological realm. Physical organic principles encompass overall conformational considerations, chemical properties, and molecular electrostatic potentials, as well as distinctly localized stereochemical, hydrophilic, electronic, and steric parameters. Understanding such interactions can provide fundamental, basic knowledge that is general as well as compound specific in its applications directed toward either enhancing the overall profile of a certain molecular display or designing a new chemical entity or NCE. Example, by effecting small molecule-driven perturbations of discrete biological processes or of overall biological pathways for the purpose of eliciting a specified therapeutic endpoint. Drug molecules can be viewed as a collection of functional groups which confer specific chemical and physical properties. Functional groups determine such characteristics as ionization, solubility, reactivity, chemical stability, and in vivo metabolic stability. Furthermore, these groups are capable of forming specific bonds with their receptors and are thus extremely important in drug activity and potency. Chemical bonding is a basic concept in medicinal chemistry and in any branch of chemistry. Small molecular displays should be thought of in terms of low molecular weight compounds that are typically of a xenobiotic origin and thus not in terms of biotechnology-derived polymers. While the latter are being aggressively addressed by other fields, it should be additionally noted that the consideration of specific details associated with the interaction of small molecular portions of more complex biomolecular system still falls within the purview of medicinal chemistry's stated focus. Alternatively, the biological realm should be thought of very broadly, that is, so as to encompass the complete span of new admit, that is, absorption, distribution, metabolism, and excretion, toxicity, related systems, as well as the traditional span of biological surfaces that might be exploited for some type of efficacious interaction. The technologies that can be deployed as tools to study these interactions at medicinal chemistry's fundamental level of understanding are, by intent, dissociated from the medicinal chemistry's definition. Presently, such tools include biotechnology-related methods such as site-directed mutagenesis, combinatorial chemistry, methods provided that the latter are coupled with knowledge-generating structural databases, and long-standing synthetic chemistry manipulations that can be conducted in a systematic manner on either one of the interacting species in order to explore surface activity relationship or SARs. In the future, medicinal chemistry should be equally prepared to exploit whatever new tools and techniques that become available to allow it to proceed more efficiently along the lines of its technology-independent definition. The discovery of a new drug not only requires its design and synthesis, but also the development of testing methods and procedures which are needed to establish how a substance operates in the body and its suitability for use as a drug. However, the role of the testing methods used in medicinal chemistry in the discovery of new medicinal compounds should not be confused with the role of quality control testing in the production of drugs in the pharmaceutical industry. In the former case, the tests are used to determine how the drug may be safely administered, whereas in the latter, they are used to monitor the quality of the drug as it leaves the production line. Drug discovery may also require fundamental research into the biological and chemical nature of the diseased state. These and other aspects of drug design and discovery require input from specialists in other fields and the medicinal chemist to have outline knowledge of these fields. Medicinal chemistry is a stimulating field as it links many scientific disciplines and allows for collaboration with other scientists in researching and developing new drugs. Medicinal chemists apply their chemistry training to the process of synthesizing new pharmaceuticals. They also improve the processes by which existing pharmaceuticals are made. Medicinal chemists are focused on drug discovery and development and are concerned with the isolation of medicinal agents found in plants as well as the creation of new synthetic drug compounds. Most chemists work with a team of scientists from different disciplines including biologists, toxicologists, pharmacologists, theoretical chemists, microbiologists, and biopharmacists. Together, this team uses sophisticated analytical techniques to synthesize and test new drug products and to develop the most cost-effective and environmentally friendly means of production. Now, another question you would want to ask is, what is the future and scope of medicinal chemistry? The field of medicinal chemistry showcases milestones in pharmaceutical research and development and features expert analysis of emerging research from the identification of targets through the discovery, design, synthesis, and evaluation of bioactive agents. As high-throughput screening results are amassed into mountains not just for efficacy data but for each of the admit parameters as well, it should ultimately become medicinal chemistry's role by definition to attempt to understand and codify these crisscrossing ranges if such data is to be effectively merged and used to either select or design the most promising preclinical development compounds. In this regard, it can now be emphasized that the common denominator required to correlate the HTS data from one pharmacological setting to that of another ultimately resides in the precise chemical structure language that medicinal chemistry has been evolving since its emergence as a distinct field, that is, SAR defined in terms of physical organic properties displayed in 3D space. This in turn suggests that within the immediate future of the new millennium, it should be medicinal chemistry that rises to become the central interpreter and distinct facilitator that will eventually allow the entire new drug discovery paradigm to become successful. This central role for medicinal chemistry may become even more critical in longer term, that is, for the next 50 to 75 years of the new millennium. Speculating that the new drug discovery paradigm will indeed mature within the next 25 years into a synergistic merger of efficacy and thorough admit HTS systems that allows for an effective multi-parameter survey to be conducted at the onset of the discovery process. The accompanying validated predictive data that will have been generated over this initial period should be statistically adequate to actually realize today's dream of virtual or in silico screening through virtual compound and virtual information. However, this futuristic prediction again depends upon the entire maturation process being able to proceed in a knowledge-generating manner. Central to the latter is again medicinal chemistry as the common denominator. For example, with time, it can be expected that just as various pharmacophores and toxicophores have already been identified for various portions of the biological realm associated with efficacious or toxic endpoints respectively, specific molecular properties and structural features will likewise become associated with each of the ADME behaviors. Indeed, work towards such characterizations is already progressing in all of these areas. Understanding the pharmacophores, metabophores, toxicophores, etc. in terms of subtle differences in molecular electrostatic potentials as well as in terms of simple chemical structural patterns will eventually allow for identifying optimal composites of all of these parameters across virtual compound libraries as long as the latter databases have also been constructed in terms of both accurate 3D molecular electrostatic potentials and gross structural properties. The most striking differences from the long-standing practice of medicinal chemistry are data reduction of huge amounts of rapidly derived HTS biological results, greater emphasis upon multi-technique chemical structure considerations, and most importantly, the simultaneous attention given to all of the admit related parameters along with efficacy and efficacy-related selectivity during lead compound selection and further design or enhancement coupled with an expanding knowledge base that offers the possibility for achieving synergistic benefits by taking advantage of various combinations of multi-agent, prodrug, soft drug and or multivalent drug strategies. From an experimental point of view, medicinal chemistry's pursuit of the efficacy displayed by the small molecules in a direct manner probably won't change much in the near term beyond what has already been proposed in terms of logical extensions from some of the latest trends. For example, even the intriguing directions that are encompassed by chemical genomic strategies, where in some cases a small molecule may provide efficacy in an indirect manner, though its interactions with biochemically modified genomics and proteomics signaling systems still reduce down to being able to exploit the same fundamental principles already cited within medicinal chemistry's definition so as to achieve the specific interaction with the modified biological surface that then serves as a mediator toward efficacy. In terms of targets, bioinformatics will certainly associate pathophysiology and individual variation with useful genomics and proteomics information so as to maintain the plethora of traditional and novel pharmacological targets well into the millennium. Web-based public efforts and commercial databases coupled with desktop programs are already being positioned to make such information readily available to everyone. Alternatively, longer term into the new millennium, gene therapy will hopefully have eradicated many of today's targets that derive strictly from hereditary gene-based abnormalities. Somewhat along these same lines, the ongoing characterization of single nucleotide polymorphisms or SNPs and the further pursuit of individually tailored therapies as presently being promoted by the new field of pharmacogenetics may also contribute toward some new targets. Web-based public and commercial databases in the pharmacogenetic and specific SNP areas, along with commercial sources for the latter are experimental technologies, are also making this type of information generally available. In the near term, the field of pharmacogenetics is likely to have its first major impacts upon refining clinical studies for late-stage preclinical candidates and upon developing improved indications, contraindications, and dosage regimes for marketed drugs and for compounds undergoing clinical study. Long term, however, pharmacogenetics should become instrumental in shaping the overall nature and subtleties of the efficacy targets that are pursued rather than the numbers of the new targets that relate to novel mechanisms that might be deployed. For example, while one can speculate that the present trend to pursue curative rather than palliative treatments will continue for at least the short term, it is likely that in the longer term a growing emphasis will be placed upon preventive rather than either palliative or curative treatments. Toward this end, pharmacogenetics should become of central importance owing to its potential to divide recipient populations into distinct treatment subgroups based upon their predisposition profiles coupled with their general admit-related drug handling profiles, wherein both sets of criteria may eventually become accessible before or shortly after birth using gene-based assays and, to a lesser extent, administration of diagnostic probe molecules. Depending upon the variability of an individual's environmental exposures, it can be imagined that in the future, pharmacogenetic profiling will be done at routinely scheduled intervals through the entire course of one's life. Current research efforts are now focused not only on discovering new, biologically active compounds using ever-increasingly sophisticated technology, but also on gaining a better understanding of how and where drugs exert their effects at the molecular level. One should not underestimate, however, that the discoveries in the 20th and 21st centuries and before represent an amazing amount of insight, determination, and luck by researchers in chemistry, pharmacology, biology, and medicine. Medicinal chemistry has evolved rapidly into a highly interdisciplinary field enriched by the collaborative efforts of experts from a wide spectrum of specialist areas, from chemoinformaticians and physical chemists to molecular biologists and pharmacologists. The field of medicinal chemistry showcases milestones in pharmaceutical R&D and features expert analysis of emerging research from the identification of targets through to the discovery, design, and synthesis and evaluation of bioactive agents. There has never been more pressure to produce potent and safe compounds faster and more efficiently. Principal themes that are included in the medicinal chemistry are drug discovery technologies, design and synthesis of novel lead compounds, computational approaches to synthesis and modeling, biophysical techniques for structural characterization, overviews of novel therapeutic strategies and emerging targets, drug targeting and delivery, ADME toxicology investigations, commentary on science, policy, economic and IP issues. The careers in this field are basic research into how various chemicals affect biological systems, drug development including formulating drugs used to treat patients with diseases, testing potential new bioactive compounds in patient populations, developing guidelines for how new pharmaceuticals will be, such as chemists at the Food and Drug Administration, that is FDA, who review new drug applications from pharmaceutical companies and the processes by which the substances are made. That was all for today's topic. Hope you have understood it. Thank you for your attention. Goodbye.